Thank you, and I thank the organizer to give me the opportunity to present our results. And today I will focus my presentation on resistance to viruses in eukaryotic phytoplankton populations. So before speaking about viruses, just a brief introduction on the hosts that we use, which are small unicellular green algae, which are eukaryotes. They belong to the genus Ostrococcus, Bathycoccus, and Micromonas. And their characteristic is that they are very small eukaryotic cells because it's the size of a bacteria, about 1 micron, even less for Ostrococcus, with a minimal cellular organization for an eukaryote, with 1 nucleus, only 1 chloroplast, only 1 mitochondria, 1 Golgi apparatus. There is no cell wall, and there is no flagella. So that's a minimum for an eukaryotic cell, but it is still an eukaryote. And Bathycoccus has a similar organization, except that there are scales, polysaccharide scales around. And for Micromonas, also the same size, more or less, but no scales, but there is a flagellum, and so that's a mobile cell. These 3 genera are very common in all the oceans over the planet. And two interesting characteristics are first that they can be grown very easily in the lab, and the second characteristic is that there are many viruses in seawater. And if you take a culture and you put a few milliliters of seawater, not filtered of course, in the cultures, you get very rapidly a lysis very often, and you have a crash of the population within 2 days around. And we characterize the lytic cycle of one of Ostrococcus viruses here, which run on 8 hours, more or less, with the appearance of a viral particle inside the cytoplasm within 6 hours, and the appearance of the first lysis after 8 hours. And the burst size is small because at every lysis there is a release of about 25 viral particles. But the cells are very small, and the viruses are rather big, at about 120 nanometers in diameter. Well, just to finish this introduction, so again, the tree, the crown of eukaryotes here, with the plants and the metazoans here, just to localize everything. And here, the prasinophytes for these 3 genera are located here in the diverge at the base of the green lineage here. And there is another set of viruses which are well characterized on green algae, on tribuxophytes, so the chlorella viruses. And interestingly, there are 6 prasinoviruses which have been sequenced and 6 chlorella viruses which have been published also. And these are all double-strand DNA viruses with big genomes, about 200 kb for this one around, and bigger for the chlorella viruses, about 350 kb. And here you have the number of open reading frames for all of these viruses. Well, and just to come back on prasinoviruses, so we'll focus on them. So here are the 6 genomes which have been published, and here you can see that there is a well conservation of the global synteny of the orthologous genes between the 6 genomes. It is well conserved, except here there is a switch for some of the viruses of Ostrococcus. Here you have the 3 Ostrococcus viruses, sorry. Here 2 Bathycoccus viruses, and here it is a Micromonas virus. Another interesting point is here the presence of 2 very big open reading frames, 2 very big genes of 3,000 and 5,000 amino acids here, which are completely unknown and which are specific to the Bathycoccus viruses. So this is something intriguing. And of course, what we did was first to determine the core genes for the 3 Ostrococcus viruses, so we can define here. Then the core genes for the Bathycoccus viruses here, and from that we could define the core genes for the prasinoviruses with about 125 genes which are common, orthologous to the 3 genera for the 3 virus infecting the 3 genera. Well, but I will not go into detail in these viruses, and will not describe in more detail these viruses, and I will focus on 2 phenomena that we recently studied in the lab, which are first the specificity of these viruses and then the appearance of resistances. So first about the specificity. So we had some indications. So we thought initially that these viruses are very and highly specific. And to check that, what we did, we used different strains of Ostrococcus that we had in the lab of the species Tori, and we checked and we tried to isolate new viruses from seawater by plating seawater on loan of these different strains. And then we could amplify by picking the lysis plagues. We amplified the virus and we checked the specificity. And we got this kind of table with because we got 40 different viruses that we isolated, and we had 13 different Ostrococcus tori strains, so belonging to this species, and all isolated in Mediterranean at different locations and at different times. So we got this kind of table with here the 40 viruses that we isolated, and here the 13 different strains of the same species, Ostrococcus tori, and we added some of the strains belonging to closely related species, Ostrococcus, but of other species, and we also added the two genera, Bathycoccus and Micromonas, to test the specificity. And what we saw is that there are some viruses which are highly specific and which are able to lyse only the strain where they have been isolated. And in contrast, we have some of the viruses which are able to lyse several strains and even strains belonging to different species. But we never saw any lysis of strains belonging to another genus. And globally, we should say that the viruses are mostly species-specific, with some exceptions that you can see here, but most of them are species-specific, but they can infect different clones of, because all these strains, of course, are clonal. Well, a second phenomena that we observed in parallel, and we started to study this phenomena, and this is more or less linked to specificity, is the appearance of resistance that we saw in different cultures. And this was observed on different clonal strains of the host. So all these strains of Bathycoccus, Micromonas, and Ostrococcus are clonal. And what we see is that when the strain is growing, that you add the virus, you have a crash of the population, but you never kill 100% of the population, even in a clonal population. And you still have resistant cells which are growing after. And even if you had viruses, they are resistant. So this is general because it is reproducible. It is observed in the three major genera of Mammillales. And interestingly, it is very stable in vitro. For example, we have two resistant strains for 2 years now, and which are subcultures regularly every week, and they are still resistant 2 years after. And to try to characterize in depth this phenomena of resistance, we decided to focus here on the system Ostrococcus and its associated virus, OTV5. And to study it, the question was, what about these resistant cells? Are they belonging to only one phenotype, or do we have a mix of resistant cells, even if initially it was a clone? And to do that, we took the resistant cells, so the resistant hosts. So we did limit dilution. We could plate the cells on soft agar, and we get colonies of resistant cells. And so we got clones of resistant cells that we could analyze and characterize. And what we got is two types of resistant cells. The first type was more or less what we expected. So there are resistant cells. So if we subculture, they restart and again, we can propagate them during more than 1 year. And there is no production of virus here. And the second type was more surprising for us and not expected really, was the same. So we have the cells which grow and it is stable and they are resistant and we can subculture them. But interestingly, they produce infectious viruses, but they are not lyse, they are resistant, but they continuously produce viruses. And we have these two types that we call resistant non-producer and resistant producers. And we did that in 3 independent experiments, and we saw that about half of the clones were producers and half of the clones were non-producer. And I will not show all the experimental details to characterize these two phenotypes of resistance, but just to summarize, For the non-producers, we check that there is a complete absence of virus in the host cells by PCR hybridization or post-field electrophoresis and so on. So we did many controls. There is no virus inside the cells. And in contrast for the producers, so there is a production of 1 to 3 infectious virus every day and per cells. And I remind you that the birth size is about 25. And we checked also that there is no integration of the viral genome inside the genome of the host. And this is very clear now. And we have so these two resistance. And as we are working in a oceanological laboratory, we're interested of the behavior of in the environment of these resistant cells. And the first question was, is there a cost for this resistance? And to do that, we tried to measure the growth rate of the different resistant cells compared to the wild type to see if there is a cost, but we couldn't see any difference in the growth rate. But this is not very sensitive. And to try to examine that in more detail, we did the competition experiments, first between non-producer and resistant non-producers and wild type here. And we could measure, by adding viruses, we could measure the remaining resistant cells over time. And we see that there is a progressive decrease of resistant cells. And every time when you do the experiment, because of course we did a mix of 50-50 of the two clones, and every time we have the wild type, which is the winner. And the second competition experiment is between the non-producers and the producers, so the resistant. And here we can measure the release of the virus, which indirectly measures the presence of the resistant producers. And every time we have the resistant non-producers, which are the winners. And so finally, what we have is the resistant producer, which has a higher cost of here compared to the non-producer, which has a lower cost, but still a cost compared to the wild type here. And with this system, we can more or less quantify the cost of this resistance. Then the second question was, what about this resistance and this phenomena in environment? And to do that, we went back to the sea. And we went back to a coastal lagoon here, which is a salty water lagoon in Mediterranean. It is linked by a channel to the open sea. And in this lagoon, we know that there are many viruses and a high concentration of viruses. So and it is close to the lab, so that's very convenient to work. And so we took seawater and we tested the viruses. We tried to measure the viruses on seawater on different hosts, so the wild types, so the sensitive cells, or the resistant producers or non-producers. And we counted the lysis plaques just here. And the question was, is there a general resistance in the environment, as we saw in vitro when we did our experiments? And so what for the wild type, so first we got what we have usually, so between 2 and 3 viral particles, infectious particles, and lysis plaque, more lysis plaque per ml of seawater. So this is what we usually get. But very surprisingly, and this was counterintuitive, and we were very surprised, and we did many times the experiment, when we did the same, when we plated the seawater, the same seawater on resistant cells, both producers and non-producers, we got between 6 and 7 times more lysis plaques than on the wild type here. And this is very surprising, and we did many times the experiments to be sure, and we have every time the same conclusion, that there are more viruses in the environment which are able to lyse our resistant cells than the susceptible ones. This was surprising, and to better characterize that, we tried to characterize the features of viruses attacking the resistant cells. So we picked up the lysis plaques, we amplified the virus in liquid, and then we tested these viruses on different host cells, so the wild type, and different clones of producers or non-producer resistant cells. And what we got here, you have the different strands, host strands where it has been tested. So the wild type here, so the sensitive cells, you have 4 clones of resistant cells non-producers, and 4 clones of resistant cells, which are producers. And we got 3 kinds of viruses. The first kind of viruses are the viruses which are able to lyse the wild type, the sensitive cells. So this is the initial virus that we had, but we find other viruses here, but which are unable to lyse the resistant cells. This is the first category. The second category here in gray, here are the viruses which are able to lyse all the cells, so the wild type, the sensitive, and the resistant cells together. And the third type, sorry, oops, I don't touch at the beginning, sorry. What happens? Sorry for that. Okay. And so the third type is are the viruses which are able to lyse all the cells, the wild type and the resistant cells. And the last category is the virus which are not able to lyse the wild type, but which lyse only the resistant cells. But if you look in more detail at the table, you see that the specificity of the virus attacking the resistant clones is not related to the type of resistance. In other words, we never found a virus attacking only, lysing only the producers or only the non-producer of this kind of categories. But there is no correlation finally. And if you quantify that, so what we got are about 60% of the viruses which are able to lyse only the resistant cells, 12% of the viruses which are able to lyse only the sensitive or the wild-type cells, and about 30% of the viruses which are able to lyse both the resistant and the sensitive or the wild-type cells. So this is what we got, and these are the data we had. And this is interesting because we are now, of course, this opens the door to more functional studies that we are now doing, because we are, of course, characterizing better and sequencing one category of each category of these viruses to compare the genomes, and maybe to have gene candidates which can be involved in the mechanism of resistance. And also we will do, of course, we are doing transcriptomics and metabolomics to check and to make comparison and to have candidates between these, to have candidates to explain and to explore this resistance. And just to summary these data, which are still preliminary, but which open the door to the exploration of the mechanistic of resistance, just to summarize this data, there are many double-strand DNA viruses which are able to lyse these Mammillophyceae algae, so there's 3 genera. The specificity is mainly at the species level, and we never found any virus which is able to lyse 2 different genera, even if these genera are closely related phylogenetically. The viral resistance is a very common phenomenon in the 3 genera. We got it every time when we do the experiments, with 2 kinds of resistant hosts, producers and non-producers, and again we got these 2 kinds every time, even if we start, I remind you, from clonal strains. It's the resistance or tolerance, because for the resistant producer we can call them tolerant cells or even chronic infection. And so for these resistant tolerant cells, it has a cost in the environment. And many viruses in the environment, this is very surprising, and we don't have a clear explanation yet. And this is we have to discuss with with people in ecology to try to modelize that. But many viruses in the environment are able to lyse resistant cells, more viruses than able to lyse the sensitive cells. And well, the virus attacking resistant clones are not related to the type of resistance. I would like to thank the people working in the team, and mainly Camille Clerici here, who worked on specificity of viruses during his PhD, and Rosen Thomas here, who worked during her PhD on resistance. Thank you for your attention.
Very interesting talk. Do you have any sense of whether any of your resistance is due to attachment?
For the attachment, yeah. So I didn't present this experiment because we still have some. It's not yet very clear, but apparently we have no attachment of viruses in both cases. So both in non-producers and in producers. So we have attachments. I said no. Sorry. No, I said there is an attachment apparently in both cases. But this is not yet clear because the values we have are highly variable and we have no significant values still, so it's difficult to say. But we are working on it, of course.
Okay, another quick question. You mentioned the sequencing, which is obvious, a nice thing to do. Do you have any sense how much variation is among these viruses from what you know already? I mean, you're going to find, I think, 1% sequence difference or less than 1%? In terms of what, when you sequence these, I mean, what kind of differences do you anticipate?
Well, this is what we are doing that. So we sequence now many viruses. So we have more than 20 sequences now which have been done. And when we do a comparison between them, they are on the same strain, for viruses infecting the same strains, they are very similar, but there are still some differences that we are looking for. Yeah.
Is there any changes between the original virus and the reproduced virus?
And the resistant, the virus which is our resistant cells. So up to now, we have sequenced only one, which is able to lyse the resistant cells, but not the wild type. So we have the sequence of it. So we have a few differences in terms on the core genes. So most of the genes are conserved, but we have at least, we have 16 genes which are present on the virus lysing the resistant cells, which are not present on the virus lysing the wild type. And on the opposite, we have 2 genes on the virus lysing the sensitive cells, or the wild type, which are not present in the other virus lysing the resistant. And most of them, except 4, are unknown or orphans. But we have 4 of them which have a function, but it's too early to speak about it. But yeah.
Okay, thank you. We have to switch to the next speaker.